in this section, we're going to talk a little bit about the scientific method, the process of science. So science is a way of knowing things. It's a way that we understand the natural world. A lot of things that we didn't used to understand and we thought were a great mystery, we understand now because of scientific processes, observations, and experiments that have told us how medicines work, why the earth spins the way it spins, what causes weather patterns, how certain chemicals react. So all sorts of things have come about by using that evidence-based process of inquiry. And that's the scientific method. That's what they're talking about here. So the scientific method, it involves, we make observations about the world around us and maybe come up with questions, things that we want to answer. We have hypotheses or educated guesses about what we think is the reason behind something that we see happening. We then run tests of our hypothesis. That's called an experiment. And then we collect data, we analyze that data, and then we can draw conclusions. So a hypothesis is sort of your guess before the experiment. A conclusion is the results accepting or rejecting that hypothesis based on a single experiment. And ultimately, scientific theories are supported by lots of experiments. So we may have the theory of relativity, different kinds of theories that you hear about in science. And they are supported by lots and lots of evidence. We don't ever say that they're really facts. We believe that there's always room where we might discover something that changes or tweaks a theory. But it is supported by a large body of evidence, many, many experiments. Louis Pasteur, a great scientist, said, where observation is concerned, chance favors only the prepared mind. In other words, you need to be ready for whatever you might see. So this is a little video, and I played in class, so I'll go ahead and play it for you.
Clearly, somebody in this room murdered Lord Smythe, who, at precisely 3:34 this afternoon, was brutally bludgeoned to death with a blunt instrument. I want each of you to tell me your whereabouts at precisely the time that this dastardly deed took place.
I was polishing the brass in the master bedroom.
I was buttering his lordship's scones below stairs, sir.
I was planting my petunias in the potting shed.
Constable, arrest Lady Smythe.
But how did you know?
Madam, as any horticulturist will tell you, one does not plant petunias until May is out. Take her away. It's just a matter of observation. The real question is, how observant were you? Clearly, somebody in this room murdered Lord Smythe, who, at precisely 3:34 this afternoon, was brutally bludgeoned to death with a blunt instrument. I want each of you to tell me your whereabouts at precisely the time that this dastardly deed took place.
I was polishing the brass in the master bedroom.
I was buttering his lordship's scones below stairs, sir.
I was planting my petunias in the potting shed.
Constable, arrest Lady Smythe.
So this is actually an advertisement for watching for cyclists on the road, but it's really interesting, and the point is well taken. And a lot of things that we've discovered in science are because of having an open mind. Sometimes we've discovered a medicine that we test for one thing, and it turns out it works for something else. And so we always want to be open to possibilities. Be careful as far as the difference between an observation, observing, and an inference. So an observation, or to observe something, you have to physically see it. To make an inference, you're drawing a conclusion based on sort of putting evidence together. So let's do a quick example. So I showed this one in class. So if we look at this boat, what do we observe? Well, we can observe that there's a hole in the boat. Another thing some people said is that we observe that there's some black stuff around the hole. There's some people on the boat. There's some people on a boat that appear to be coming towards the camera. The boat is definitely in water, and the water seems to be near some land because we can clearly see land in the background. Now, inferences. There was an explosion. There might have been an oil spill. The people on the boat swimming, going away are people that were rescued. We don't know for sure any of those things. Those are all inferences based on drawing conclusions, based on putting our evidence together. Here's another example. So obviously we see a guy. What do we observe? He's on the ground. There's a bicycle on top of him. There's water around him and in the air. He's holding an umbrella. Inferences. Maybe I could infer it's possible he fell because the road is slippery and he didn't have good tires, so he skid. Maybe he fell because he lost his balance because he was trying to hold the umbrella because of the rain. Maybe he was hit or bumped by a car passing by because it appears that he's in the street. So these would be inferences, but not observations. And one more cute little inference. So we can observe that there are heads in this rock. But do you know what's on the other side? Maybe this. Maybe they're posing for the picture. All right. Observations and hypothesis, sort of putting it all together. An observation, the flashlight doesn't work. Question that we have, why doesn't the flashlight work? Well, we might come up with multiple hypotheses. Here's two sample ones. Maybe it doesn't work because the batteries are dead. Or maybe it doesn't work because the bulb is burned out. Two possible reasons why the flashlight doesn't work. Now we would do our experiment. I predict that if I replace the batteries, the flashlight will work. My experiment, I add batteries. Let's say that doesn't work. Now, my second hypothesis, I'll try that. Maybe the bulb is burned out. I will replace the bulb. In this case, we have the results of both tests. The flashlight did not come back on when we replaced the batteries, so that hypothesis would be rejected or contradicted. But the hypothesis that the bulb was blown out, now the light turns on, so that must have been the problem, and so that hypothesis would be supported. All right, so the main requirement for a scientific hypothesis, and this is important, make sure you know this, is that it has to be testable. You can't have answer questions about things like ethical dilemmas. You know, is communism evil? There's no way to really test that. That's not a scientific question. So whenever we come up with a hypothesis, the flashlight is out because the bulb is burned out. Is that testable? Yes. We can actually set up an experiment, replace the bulb, and then look at the results. All right, so when we do set up an experiment, you need to know the parts, and you've probably learned these before. So one of them, the thing that you change or manipulate in the experiment, is called your independent variable. This is the one that you control. This is the test that you're running. And then what you measure, in other words, the data that you collect in your experiment, that's going to be the dependent variable. In fact, some books also call this the responding variable because it's the response to the change that was made. So the responding variable. A lot of times when you collect data in a data table, for example, let's say that we were doing some kind of a data table where you were adding different amounts of fertilizer. Handwriting's really bad here. Fertilizer, and maybe you were going to measure how tall the plants were. Well, your independent variable would be what you were changing. So maybe I have 0 milligrams of fertilizer. Whoops. 0 milligrams, 5 milligrams, 10 milligrams, 15 milligrams, 20 milligrams. So the amount of fertilizer I add, that would be my independent variable. I put IV for independent variable. What am I going to measure? The data that I'm going to collect, how tall the plants are maybe after a week or something like that. This is going to be my dependent variable because it depends on if they respond to that change, the independent variable. A controlled experiment always compares an experimental group, the group that receives the change, to a control group. So for example, for this fertilizer experiment, the experimental groups would be all the ones getting these different amounts of fertilizer. And then our control group would be the one getting no fertilizer, because that would give us a comparison. If fertilizer does make plants grow taller, then the plants receiving the different amounts of fertilizer should be much taller than our control group that was left alone. Keep in mind, a control group is not left alone in the sense that you just ignore it. It receives everything every other plant receives. The only difference is the one thing, the one variable, the fertilizer. So that control group plant would still get water, it would still get the same amount of light. You want to treat it the same in every way except for that one change. And that's what this is explaining. This is actually explaining, so there was an experiment done. This is just a little example that the book gave. They saw there were two groups of mice. So this says the use of control and experimental groups can demonstrate the effect of a single variable. In this example, researchers found that mice models that did not match their habitat had higher predation rates than camouflaged models. And then they explained that human hypothesis, we can test like drugs, for example, with clinical trials and observations. This is the, oops, sorry, I'm going to skip that. This is the mouse experiment. So what they discovered was the mice on the beach were almost always white and gray, and the mice that lived in the grassy foresty area, they were always brown. So they wondered, why? Why are the brown mice, and these areas are right next to each other, by the way, why are the ones in the inland population brown? And their hypothesis was that it was predation, in other words, predators. That the white mice, or the grayish ones, were camouflaged against the sand. And that if a brown one happened to go into that area, or a brown one was born, genetically, a brown one popped up, it would be less likely to survive. And so they actually did an experiment where they put fake models of white mice and brown mice here in this area. So imagine they have little fake mice. It's a terrible mouse. And they have white ones and brown ones. And then they did the same thing here. They put some white ones and brown ones. And they looked to see how often did the model mice, the fake mice, get attacked. All right, and this is their results. So what they basically found was that on the beach, oops, sorry about that. In the beach habitat, the camouflaged ones, which would be on the beach habitat, that would be the mice that were light in color, 2 got attacked. The mice that were brown in color, 5 got attacked. So 71% of the attacks that happened were on brown mice on the beach. In other words, the ones that were not camouflaged. And then they found the same thing inland. In the inland area, they found that 5 brown mice got attacked, 16 white mice, models of mice, they were fake. 16 white mice got attacked. In other words, 76% of the attacks that happened were on white mice, the ones that were not camouflaged in that area. And so from that, they would be able to draw their conclusions. All right. Now, checkpoint question. In some studies, the researchers want to match factors such as the sex, the age, and the general health of the subjects in the control and the experimental groups. In other words, if we're testing a drug on a bunch of people, maybe we want to test it on all males that are all 15 years old. They all have similar diets. They all have healthy blood pressure and all that kind of stuff. Why would they do that? And the answer is because those are called controlled variables or constants. They're trying to make sure that any change they see, like if the drug is supposed to, let's say, make them taller. We want to know if they get taller, is it specifically because of the drug or is it because of something else in their environment? And so by making every group, the group that receives the drug and the group that doesn't receive the drug, making them as exactly the same as possible, we can hopefully make the conclusion if the one group gets taller that it's because of the drug and not because they already ate more food or they slept better or, they came from a different area of the country or they were different ages. So we would want to make our groups as cohesive as possible. All right. So that's a little bit about the scientific method. In the next lecture, we'll talk a little more about making graphs of data and the proper format for doing that.